Hello, everyone, and thanks for joining us for the Cell and Gene Therapy 2023. I'm George Hammond, and I'm here to moderate today's event alongside my colleagues Caroline Clark and Princess Senate. Before we start the talks today, I just have some general information and a few tips for getting the most out of the event. Firstly, as well as the fantastic lineup of speakers we have for you, I'd like to point out the networking areas on the platform, which you can use to chat with other delegates throughout the day. On the left-hand side, you will see the People tab, where you can access a list of attendees that you can contact. From here, you can message them privately, and all your ongoing conversations will appear in the chats area. I encourage you to get involved and take this opportunity to speak to some of the over 1,500 delegates that have registered for this event. If you are not sure who you would like to chat to, we encourage you to use our participant matching feature to match with attendees who share similar interests to you. Just navigate to participant matching located in the left-hand menu and fill out a brief questionnaire. Our software will then do all the hard work for you and match you with other attendees with similar interests and goals. In the list of matches, there will also be a percentage match, which indicates how close of a match your response were to the attendees in the list. Please remember that we encourage debate and discussion and welcome all of you to use our networking services to introduce yourselves to the other attendees. But we do not permit the overt solicitation of products or services and reserve the right to block any user who infringes on this rule. On a similar note, all of today's talks offer the ability for you to engage with our speakers via the Q&A session at the end of each talk. I encourage all of you to ask the questions, as this is the best opportunity to get live answers from our panel of experts. We will explain how you can ask questions at the start of each presentation. It's also incredibly important to say that this event was only made possible thanks to the generosity of our sponsors and partners, particularly our platinum sponsor Agilent. In the Literature and Resources section, you can download free educational pieces from our sponsors and partners, including application notes, white papers, and guides focused on cell and gene therapies. For more detailed instructions on how to make the most of the features available to you at this event, please navigate to the event home and watch the overview video at the top of the page. We would like to ask if you could please refrain from video, audio, or photo recording of all talks. Many of our speakers have kindly agreed to share their slides with you, and these are available within the literature section on the platform. Some of our speakers will have included their contact details in their presentations. If they do, please feel free to reach out to them directly. If you'd like to contact the online symposia team at any point, please contact us via the Technical Help tab at the bottom of the navigation bar on the left. And with that out of the way, I will now move on to presenting our opening keynote session for the event. The precision and flexibility of homologous recombination-based genome editing. I'm pleased to welcome our first speaker, Matthew Porteus, as your keynote presenter. Matthew is a Satarja Chuk Professor of Definitive and Curative Medicine and a professor in the Department of Pediatrics at Stanford University. His primary research focus is on developing genome editing as an approach to cure disease, particularly those of the blood and most notably sickle cell disease, but also of other organ systems as well. His goal is to combine his research and clinical interests to develop innovative curative therapies. A very warm welcome to you, Matthew. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during this presentation. If you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. Without further ado, I will now hand over to Dr. Porteus to begin his presentation.
Thank you, George, for that kind invitation, and thank you to the organization for inviting me to give this keynote talk describing our work developing homologous recombination-based genome editing as a potential new way to treat patients. I have been lucky enough to be involved with some biotech companies, but almost everything I talk about today will be work that has come from my academic lab at Stanford. So I was once upon a time a history and science major as an undergraduate, and so my brief foray into history is to just remind people about the importance that new technologies have in improving the lives of people around the world. Ranging from the discoveries in the 19th century of the importance of sanitation and antisepsis and anesthesia, the development of vaccines in the 18th and 19th century, and of course, the importance of those we were all reminded of over the last 3 years. And now we have small molecule therapies, biologics, and even nucleic acid therapies. But fundamentally, one can make an argument about vaccines. These are generally interventions that, when administered to a patient, degrade over time. But what we're at the beginning of is, I think, an explosion in medicines of so-called living drugs that, instead of degrading over time, expand and respond to the body over time. And we are hopeful that these so-called living drugs will be able to address the unmet medical needs of patients who prior technologies have been able to address. And to demonstrate what I mean by a living drug, I show an experiment in our lab in which we took human CD34 positive hematopoietic stem and progenitor cells, the cells that are going to give rise to all of our blood, and we labeled them with a luciferase gene and transplanted them into an immunodeficient mouse in the left femur. You can see at 3 days post-transplant, the cells, as measured by light, are just localized to where we put them. But instead of degrading over time, you can see the light, as demonstrated in the blue, the pseudocolor plot, grows over time. And this cellular drug migrates to all parts, all bones of the body that blood is made in. And what this underestimates is that the progeny of these stem cells, including red blood cells, platelets, neutrophils, and macrophages and microglia, have migrated into the different tissues of the body. And so you have a drug now that is systemic in nature, and in the case of hematopoietic stem and progenitor cells, has been demonstrated through bone marrow transplantation to have lifelong durability. Combining this concept of living drugs is the idea of precision genetic engineering using genome editing. And the implication of genome editing is that you can modify the sequence of the DNA of a cell in the same way that one can modify a document on your computer. And that could include correcting typographical errors. It could mean adding a new sentence into a paragraph that changes the meaning of that sentence. It could mean replacing one part of the sentence with another part of a sentence, or replacing entire sentences. And finally, it can mean adding clauses to sentences so that the sentence now has new meanings. And this is what, this flexibility is what we really want to be able to do, to not only address underlying pathologic genetic causes of diseases, but to enable us to engineer cells with new therapeutic properties, the concept of synthetic biology, to give cells to patients to treat both rare and common conditions. And we use, the oldest form of genome editing, which is to engineer a nuclease. And I worked with zinc finger nucleases and tau effector nucleases and homing-end nucleases. But we now use the CRISPR-Cas9 system, not because it's the latest and greatest, but in our hands, it is the simplest to design. It has the highest reproducible activity, and in fact, extremely high specificity. And by designing a CRISPR-Cas9 guide RNA complex to the Cas9 molecular scissors, we can create a break in our target site of interest. This break then gets repaired by the cells in a number of different ways. If it gets repaired precisely by non-homologous end joining, the target site of interest is recreated, and the CRISPR-Cas9 complex is so active, you'll create another break. And what you end up is in this iterative cycle of break and repair until either the site gets inactivated through a small insertion or deletion, or if one provides a donor template, a molecule, a DNA molecule that has homology to the site of the break, the homologous recombination pathway will insert the sequences from the donor into the genome. And so here I've shown that a target site of interest could be converted into a target site of great interest. And great can represent all of those changes that I showed before, from single letter changes to the insertion of full sentences. So if we convert those sentences into genetic manipulations, they're highlighted here, where you change a single nucleotide, you insert a copy of a gene back into itself, you insert a whole gene cassette, including a promoter, into a safe harbor, You can replace one gene with another gene. You can insert one gene at the start of a gene, or you can insert a gene at the end of a gene so it gets expressed along with the first gene. And in my talk, I'm going to give three stories of how homology-directed repair or homologous recombination can achieve all of these with very high frequencies in hematopoietic stem cells and T cells, among other cell types. The system we use took years in development and is summarized here. It is a system in which we manipulate cells outside the body because the cell is the drug. And we put the cells into cycle because that is when the homologous recombination machinery is most active. We deliver the CRISPR-Cas9 as a purified Cas9 protein, which is, we use a high fidelity variant that was discovered by IDT. And we complex that to a single synthetic stabilized guide RNA, or an S-cubed guide RNA. And this complex is delivered via electroporation. We then deliver the donor template on a AAV6 virus, because AAVs have evolved to deliver nucleic acids to the nucleus of cells while being minimally detected. I'll show you, however, that there is some detection. And using this system, we're able to achieve targeted integration frequencies, or HDR frequencies, of up to 80% in essentially every cell type we can grow in the lab. So the first example I want to show is to change an A to a T. And this is applicable to the disease sickle cell disease, in which a devastating autosomal recessive disease, perhaps the most common serious genetic disease in the world, in which every patient contains the same single nucleotide variant, a T replacing an A, in codon 6 of the beta globin gene. This leads to a single amino acid change from a charged amino acid, glutamic acid, to a polar amino acid, valine, causing the hemoglobin tetramer, which normally is free, to polymerize, converting red blood cells into sickle cell shape that gives the disease its name. These stiff sickle cell red blood cells occlude the blood vessels and prevent oxygen from getting to tissues, including the bones, leading to painful bony crisis shown in the painting on the left, but includes strokes, progressive kidney damage, progressive heart damage, progressive lung damage. And in the US, the median lifespan, despite decades of improvements in supportive care, remains in the mid-40s. In Africa, where there's over 300,000 births per year, the median lifespan is in the first decade of life. So a large team of people, and first a publication in 2016 and then a follow-up publication in 2021, showed that we could design a CRISPR to cut the beta globin gene, a donor template that would convert the pathologic T back to the pathologic A, as well as a donor that had silent changes to prevent indel formation following correction and to facilitate high frequencies of HDR. And using the system in the laboratory, shown in green, is that we can achieve 60 to 80% gene correction in the stem and progenitor cells from patients with sickle cell disease. When these cells are turned into red blood cells in vitro in a tissue culture assay, the unmanipulated cells, labeled here in mock, create essentially all hemoglobin S with a small amount of hemoglobin F. But after the gene correction process occurs, we convert essentially all of the hemoglobin S to the green non-pathologic hemoglobin A. There's a slight increase in the amount of hemoglobin F, the importance of which we're still trying to understand. But hemoglobin F is a variant that counteracts hemoglobin S. We were able then to take this laboratory process and scale it up, as not surprisingly, the overall efficiency, as it scaled from a couple million cells at a time to several 100 million cells at a time, the efficiency decreased somewhat. And you can see at first in run one, we were very good, perhaps beginner's luck at scale. And then over time, we got slowly better and better as we understood the process at scale, as well as making subtle changes in the manufacturing process to increase the frequency of the green. When we looked at the final process and analyzed where was the cost of manufacturing come from, we found that 50% comes from the consumables, 20% comes from the suite, 20% comes from labor, and approximately 10% from the analytics and release testing. When we break down the consumables, we find that most of the price of the consumables comes from the cytokines that are used to grow the hematopoietic stem cells in culture. What we can see is that while AAV is a very expensive gene therapy reagent when given to people in the body, in the ex vivo setting in the cell manipulation space, it is actually quite low on the cost of goods because we use relatively little AAV to achieve the gene correction frequencies I described. I bring this up because it is not just the discovery science, but the manufacturing and process engineering that is one day going to be so important in bringing these living, genetically engineered living drugs to more patients than just the few with serious genetic diseases. We then performed a series of assays to assess safety and toxicity. These assays were done before the FDA released a draft guidance for genome-edited medicines, but they generally follow what the FDA guidance recommends, which is to explore whether indels or mutations are created at potential off-target sites, and they recommend identifying the potential off-target sites using orthogonal methods. They ask to look, do the cells develop any structural variations? And there are increasingly sensitive and easier assays to use. And then they want to see measurements of functional toxicity. Do the cells behave as one would expect in vitro and in vivo assays? So we carried out all these assays, and they were published in the Science Translational Medicine paper in 2021. And our IND was cleared, and this IND was then licensed to Graphite Bio, and Graphite Bio then took over the role of sponsoring the clinical trial. In August of 2022, Graphite made a public announcement that the first patient had been dosed with her own autologous hematopoietic stem cells that had gone through the gene correction process I just described. The drug now has a name called Nula-beglogene autoged-tem-cel, or Nula-cel. And the infusion went without complications. Now, in January of 2023, the company announced that the patient still was requiring red blood cell and platelet transfusions. Following an autologous bone marrow transplant, it is normal for patients to require platelet and red blood cell transfusions. However, this patient's requirements was going longer than expected. And so the company made, as a public company, it released this information as it was required to by the SEC. Since then, the patient, however, had no MDS, was at home and doing well, and the patient continues to improve and has not reached a steady state. And we hope that in the near future, a steady state will be reached, and we will be able to publicly report on the full details of what happened. I will say, however, that instead of treating a second patient, we know that there was need to make the manufacturing process better. And so the company put the clinical trial on a voluntary pause. The FDA has not put the trial on a hold. And when we look back at our process, we are reminded we can see evidence where we might need to improve the process. So in the blue dots on both the left-hand chart and the right-hand data are the functional characteristics of cells that have been unmanipulated. And in vitro colony forming assay, you can see blue, and in green are the gene-corrected cells. And you can see on the left-hand side that there are fewer colonies in the green products than the blue products, although there's overlap. And similarly, in the mouse engraftment experiments, you can see that the blue the mice that received unmanipulated cells had, in general, higher levels of human cell engraftment, although there was still, even among healthy donor cells, there was 2 logs different. You can see 1% to almost 100%. And the manipulated cells, on average, had lower levels of engraftment, but significantly overlapped with the blue. Nonetheless, given what happened in the patient, this gives us a target for how we should improve the process, which is, can we make green come closer to blue? And we and others have noticed that when you expose hematopoietic stem and progenitor cells to AAV6, while there is minimal transcriptional changes, there is some evidence of an activated p53 stress response as measured by increases in the transcription factor p21. And on the left-hand side, what you can see is this is dose responsive, so that the more AAV you add from 625 to 2500, the higher levels of p21 are activated. Similarly, as we look at that colony forming unit assay, we can see that the more AAV you add to the cells, the much fewer colonies you get. And if you can reduce the MOI from 2500, which was used in the clinical trial, to 625, you can see that now the number of colonies approaches the number in wild type, in unmanipulated wild type cells. So this gives us a target. So how can we get there? Well, one way is perhaps by utilizing a molecule to alter the way cells repair double-stranded breaks, and specifically using a small molecule inhibitor of the NHEJ pathway. There's a number of these inhibitors, including a drug from Merck, AstraZeneca, Vertex, Bayer, and others. And Sridhar Selvaraj, shown here in the lower right, identified the small molecule inhibitor from AstraZeneca as one that had functionally the best activity, potency, as well as the least toxicity. One can see is that these contemporary DNA PK inhibitors have biochemical profiles suggesting high specificity for the target protein kinase and low specificity or low or minimally inhibiting the related kinases such as ATM, ATR, and the PI3 kinases. And what Sridhar showed is that in the beta globin example, by adding the small molecule, we increase the amount of HDR, shown in white, and decrease the amount of indels, which is good, but what I think is even more important is shown here, in which by adding the DNA PK inhibitor, not only did we increase the amount of HDR in this experiment at the same MOI of 2500, it went from 46% to 75%, but it allowed us to lower the MOI to 600. And at an MOI of 600 with a small molecule inhibitor, we had 58% HDR, which exceeded the HDR percentage at the higher MOI. So we got 2 positive effects, lowering the MOI with higher HDR. And this results in a reduction in the P21 response, that downstream marker of P53, to a range that is seen between unmanipulated cells and cells that have been just exposed to the CRISPR-Cas9 RNP complex. We use the CRISPR-Cas9 RNP complex as a target because several companies have now shown that if you manipulate hematopoietic stem and progenitor cells with the RNP alone, that the cells engraft well. So we believe if we can reach the biologic specs of RNP alone, our cells are likely to engraft better. And using this manipulation of DNA repair, we can achieve cells that have this range of P53 activation. So as we move from the lab to the patient process, I want to point out that the initial discovery was a lot of work and extremely important. But since then, there have been multiple other activities that have been essential to getting from the lab to a patient, including the development of analytics of all varieties and establishing critical quality attributes, IND enabling toxicology and biodistribution studies, process development, demonstrating reproducible clinical scale manufacturing, establishing a reliable supply chain for a range of different molecules, then being able to recruit your first patients. And I think what I've learned is then to prepare and expect and have the capacity to learn from those first few patients so that you can give the next patients an even better product. With that, I'd like to just move on then to the second example. And this is to use homology-directed repair, genome editing, for cystic fibrosis. Cystic fibrosis is also a common serious genetic disease, not quite as common as sickle cell disease. There are estimated to be 30,000 patients in the US, and I'll show you later that there's estimated to be maybe 200 to 300,000 patients worldwide. Unlike sickle cell disease, while there is a predominant mutation that causes cystic fibrosis called delta F508, there are also thousands of mutations that are scattered throughout the genome. This disease has been transformed by the development of small molecule modulators of the cystic fibrosis channel, but there still remain a large number of patients for which these modulators won't work. And that has been highlighted by work done by Shriram Vaidyanathan and Zach Sellers at Stanford, who looked at the genotypes of causing cystic fibrosis from different demographics and found that in patients from South Asia, the frequency of modulator non-responsive genotypes, it was not 10%, but was closer to 40%. This complemented work from Megan McGarry and Susanna McCauley, who similarly showed in the Black population that the genotype frequency of patients non-responsive to modulator therapy was much higher than what had been described in Caucasian populations, and again, approached 30% or more, which means that if you look globally, that the percent of patients predicted to be not eligible for modulators might be up to 40% of all patients, which Shriram and Zach estimate may be as high as 300,000 patients around the world, because this disease may be underdiagnosed in certain parts of the world. So our concept then is to think about, can we use cells to treat this airway disease? Now, one of the major sites for morbidity and a source for chronic lung damage is the sinus of patients with cystic fibrosis. Many patients develop chronic sinusitis. And we thought that the sinus would be a point for us to test the use of cell-based therapies to treat this disease, because the sinus is accessible, it's easily monitored, and if something did go untoward, we could intervene in a relatively simple fashion. So the idea is to take airway basal cells, the stem cells of the airway from patients with cystic fibrosis, correct their underlying mutation using the RNP AAV6 system I described to you, purify these cells, and then instead of just squirting them back in the sinus, embedding them into a matrix such that when they're surgically implanted back in the sinus, they are allowed to engraft. And I'll show you the progress we've made. Now, the genetic correction strategy we're using is one in which we take the full wild type CFTR cDNA and insert it back at the endogenous start codon. In this way, the channel will be expressed by the endogenous regulatory elements and be expressed at the levels and in the cell types needed to correct the underlying deficiency. This process has been validated in other diseases, such as in SCID-X1, and the ability to insert large cDNAs was shown by Rasmus Bach, who now runs his own independent lab at Aarhus University. And so Shriram took these two prior pieces of work and developed a gene correction system in which he could insert the full CFTR cDNA at the endogenous start codon and accompanying that with a inert cell surface receptor that would allow us to enrich for the cells that had undergone the gene correction process. So the overall process was, because we were doing this double AAV system, was lower than what we see when we just use a single AAV. But because of the selectable marker, we could use magnetic bead enrichment and enrich the cell population up to 60 to 80%, with 60 to 80% of the cells having at least one allele corrected. And when these cells were put into an electrophysiologic assay using air-liquid interface cultures, we can see that the electrophysiologic properties of the population of cells undergoing correction in red overlaps, 100% overlaps with the electrophysiologic properties of non-CF patients compared to the original controls, where the electrophysiologic properties were significantly decreased because of the underlying mutation. So now this gets to the challenging part, is how are we going to give these cells back? So the first thing Shahram did is explore a variety of matrices to identify one that these basal cells would grow in. And he identified fibrinogen and biosilk as possible matrices. And we decided to proceed with fibrinogen because this is a scaffold that is already used surgically. So we thought there was a precedent here. So we then embedded ourselves in fibrinogen, and Dawn Bravo was able to show that when she took human gene-corrected basal cells, embedded them in fibrinogen, in this case, actually, they're not gene-corrected, they're marked with luciferase, and embedded them in fibrinogen and transplanted them into mice sinuses of immunodeficient mice. The mouse sinus had been debrided of its epithelium in various different ways. All of them worked. You can see long-term persistence of these human cells in the sinus, as shown by the light emissions from bioluminescent imaging. And histologically, when Dawn took down these mice, she could see patches of human epithelium embedded within the reconstituted mouse epithelium. So the human epithelium, in this case, in this panel is shown in green, surrounded by the mouse epithelium shown in white. So we had a pre-IND meeting in December of 2022, and they outlined, and we came to an agreement on our path forward in terms of process development, safety and toxicology, and biodistribution studies, both in the mouse and the rabbit, and the synopsis of the clinical trial design. And I think in summary, what I want to point out to this audience is to think that as we combine cell-based manipulations with devices or matrices, we can think about transplanting using cell-based therapies for tissues and organs which have been historically thought not to be amenable to cell-based therapies, giving us another iron in the fire as we think about how we might be able to use the genome editing, powerful genome editing tools to treat patients with underlying diseases. And then the last vignette, I'd like to describe my collaborative work with my colleague here at Stanford, Alice Bartagna, and that is to use genome editing in combination with a novel way of performing hematopoietic stem cell transplantation using haploidentical grafts. And in this approach, we're going to use genome editing to insert a CAR gene into the track locus. And in this way, the CAR will be expressed and the TCR will be inactivated, creating a CAR positive, TCR negative T cell. And as I said, this has been a collaboration with Alice Bartagna and was led by Volker Wiepking, who is now a medical director at Genentech Roche, and was published in Hematologica in 2021. And the platform it's built on is this haploidentical transplant platform in which haploidentical hematopoietic stem and progenitor cells are depleted of the GVHD causing alpha beta cells, as well as B cells that can cause a post-transplant complication called EBV PTLD, leaving behind NK cells, gamma delta cells, and hematopoietic stem and progenitor cells that are then transplanted into a patient. What our idea is to supplement this haploidentical transplant with post-transplant CAR T cells to enhance the anti-leukemic effect of the original haploidentical transplant. But if we give a large dose of haploidentical T cells that contain an intact TCR, that would cause GVHD. So as I said, we want to create an anti-leukemic CAR that has a TCR knockout. And the way we do that is we take the alpha beta cells that were originally depleted and then run them through the genome editing process I described. In this way, they might be given back to the patient in a way that does not cause GVHD, but because they're autologous to the incoming donor cells, the incoming immune system, they would not be rejected by the host immune system. So they would have the potential for longer durability than what we're seeing in the allo CAR T products that are being given. So we take our CD19 CAR, knock it into track. We attach a selectable marker, not a selectable marker. It is a selectable marker, but we attach a cell surface marker to allow us to easily measure this. And what Volker finds is that by optimizing the culture conditions and the genome editing conditions, he can get 80% knock-in of the CAR and leaving behind just a small number of TCR positive cells, approximately 2 to 4%. But then by taking this population and repassing it over an alpha beta depletion column, we can reduce the number of TCR positive cells to 0.02%. So essentially a drug product that now has essentially no TCR alpha beta positive cells. You can see here the reduction, the over 2 log fold reduction in TCR positive cells after a second column. And finally, these cells maintain their activity. And when you administer in NALM6 cells as a CD19 positive to lymphoma to NSG mice, and then treat those mice with doses of the genetically engineered CAR T cells, you can see at a reasonable dose that the mice are cleared of their tumor up to 13 weeks, at which point the experiment was terminated. So where do we go from here? Well, first, we'd like to develop a closed system manufacturing. And the second thing is we wanted to incorporate an ability to get rid of the cells. Because these cells are made from healthy donor T cells, and because they're going to be autologous to the new immune system, they have the potential to have prolonged durability and the potential to cause lifelong B-cell aplasia. So what we wanted is if we a mechanism by which to get rid of these cells if we thought that they were no longer needed. And the system that we're incorporating is one that Volker also developed in the lab, which is to use genome editing to develop oxytrophy. And this was published in collaboration with James Patterson in Nature Biotechnology. And the concept is the following, which is that we have multiple metabolic pathways in which there's a key enzyme that converts substrate into product. If you knock out that enzyme, the metabolic pathway is disrupted. And the only way the pathway can be rescued is to give the downstream product as a nutrient. And this is known as oxytrophy. And the pathway that James identified was a pathway involved in nucleic acid synthesis, the pathway of generating nucleotides. And the key enzyme is an enzyme called uridine monophosphate synthetase, or UMPS. And it turns out that there are a small number of patients who are born with defects in UMPS, and they have multiple problems because of their inability to synthesize nucleic acids. However, if they are giving oral uridine, just a simple nutritional supplement, they are able to rescue their underlying defect because we are rescuing the downstream. We're rescuing with a nutrient the downstream product of the UMPS enzyme. So Volker showed that if he made knockouts in UMPS and did not provide supplemental uridine, that the cells over here on the right did not grow very well, whereas the wild type cells grew just fine. But as you added supplemental uridine to the culture, these cells were rescued. Now, there's not complete rescue, but the cells are rescued well. And so how might we use this? Well, we could use this auxotrophic metabolic control in a number of different ways. But the way I'm going to show is how we could use it to control an engineered T cell product. And so Volker took healthy T cells, knocked them out with UMPS, transplanted 10 mice or actually 8 mice with T cells, and grew those mice with uridine in the diet. And then once he saw adequate levels of human T cell engraftment, he randomized the mice into one group in which they were taken off the uridine triacetate supplementation, and another group shown in orange in which the uridine triacetate was maintained. And what you can see is in the mice that stopped receiving the nutritional supplement, the T cells essentially disappeared and did become completely inactive because they had no way of proliferating. So we believe that there is now a possibility of combining this auxotrophic cellular control method with CAR T manufacturing to generate a controllable CAR T product that we could administer post-transplant to increase remission rate and yet not cause long-term B-cell aplasia. So to conclude, what once could only be drawn on a chalkboard can now be executed in stem cells with high specificity and efficiency. And I showed you how using homology-directed repair, we can achieve a number of different manipulations in hematopoietic stem cells, T cells, but we're also able to achieve this in iPS cells, B cells, mesenchymal stromal cells, neural stem cells, keratinocytes, and a large number of cell types. Hope I've convinced you that while HR-based genome editing is not the flashy new kid on the block, it still remains the platform that has the greatest versatility and data I didn't show is actually allows you to recapitulate natural human genetic variants. Showed you some of our work around technological improvements, and they will continue. And I spent a little time talking about the costs of manufacturing and the need to continue to drive innovation to lower costs so that one day these living cell-based drugs can be accessible to patients all over the world. I want to thank you for the invitation to be the keynote speaker. I get to work with a fantastic group of undergraduates and graduate students and technicians and postdocs and senior scientists in my lab. I get to work with people in my pediatric bone marrow transplant division, our GMP facility. And then I have learned over the last few years of the importance of asymmetric expertise and collaborations, and have been really fortunate to collaborate with academic and biotech companies around the world where they bring one thing to the table and we bring another, and out of that comes very successful collaborations. And to end, so some pictures of the lab. The other day, we were chatting over lunch and realized we had representation from 6 of the 7 continents of the world. The only continent not represented in the lab is Antarctica, and that prompted the lab to buy a penguin to represent the 7th continent of the world. So with that, I would like to thank you for the invitation to present, and I will finish and be happy to take any questions. Thank you very much.
Thank you so much, Matthew. That was a fantastic talk to kick the day off with. We're likely to have lots of questions from the audience today, and I'm aware of the time we have remaining. I can see that we have received some questions already, so I'll move straight onto these now. So your first question, Matthew, is what do you see as the greatest barrier to developing genome editing to treat disease?
Yeah, I think that probably there's no one greatest barrier. I think one of the exciting things in genome editing is we increasingly have different tools in our toolbox. What I mean by that is we don't simply have a toolbox with 7 different hammers, but we now have a toolbox with a hammer and a screwdriver and a wrench. And I think one of the challenges we face is to think which tool is the right one to use for which disease. Instead of thinking that one tool is the right tool for all diseases, I think this toolbox allows us to tailor. I think the second thing is the engine of discovery is strong. Every week, every month, every meeting, there are new technologies being developed. However, I think the translation of the discoveries in the lab to patients, there needs to be more innovation and drive at this still very costly process that requires millions, if not tens of millions of dollars to do. And we somehow need to streamline that process so that these technologies, hopefully designed appropriately for the patient population in need, can be tested in patients more rapidly.
That's perfect. Thank you for that, Matthew. Unfortunately, we only have time for one final question today, which is going to be, isn't using AAV going to increase the cost of goods of manufacturing?
Yeah, that's a great question. You know, there's the as I talked about in the talk is that AAV is an incredibly powerful gene therapy vector, and is the vector that is being developed to deliver genes to a wide variety of tissues in the body. The doses, however, that are required to do that in an in vivo setting are extremely high. And the manufacturing capabilities to make those high doses are getting better and better. But when we're using AAV in the ex vivo setting, we use a minuscule amount compared to what is used in the in vivo setting. And so therefore, as I showed, the AAV is not a driver of the cost of the manufacturing. It is rent, facilities, people, and cytokines that are driving our costs. And so it's important to think about the vector as it relates to how it's being used, and not necessarily just to categorize something as expensive or cheap in a global way. So I hope that answers the question for the audience and for you. So again, thank you.
That's perfect. No, it does, and you explained it incredibly well. Thank you so much, Matthew. Unfortunately, that is all that we have time for today, but it's been an absolute pleasure speaking with you, and we just want to thank you for sharing insight into some of the current research you and your team are carrying out at Stanford. Any questions that we didn't have time to get to will be sent to the speaker following the event. As of all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. And that just leaves me to say thank you once again, Matthew, and we will be back soon with the next talk.
Thank you.